This video and its supplemental guide summarize methods developed at the EPA Office of Research and Development, Narragansett Laboratory for estimating the chronic toxicity of marine or estuarine effluents and receiving waters on the gametes of the sea urchin Arbacia punctulata. Sperm cells are exposed to effluents or receiving waters in a static system for 1 hour. Then, sea urchin eggs are added for 20 minutes of static exposure. The test measures the effective exposure on fertilization. The methods demonstrated in this video and described in the accompanying guide are detailed in the EPA Methods Manual, Short-Term Methods for Estimating the Chronic Toxicity of Effluents in Receiving Waters to Marine and Estuarine Organisms. Obtaining and maintaining sea urchins. Adult sea urchins can be ordered from commercial biological supply houses or collected from uncontaminated sites along the Atlantic coast. Keep male and female animals in separate tanks. A 20-liter aerated tank can hold about 20 adults. Allow filtered seawater to flow into the tank at a rate of 5 liters per minute. The temperature in the tank should be maintained at 12 to 18 degrees Celsius. Sea urchins are fed kelp from the species Laminaria gathered from Atlantic coastal waters or ordered from commercial supply houses. Supply the urchins with ample food, renewing the kelp each week and removing decaying kelp as necessary. Every 1 to 2 weeks, empty and clean the tanks. Healthy sea urchins will attach to kelp or aquarium walls within hours. Any unhealthy animals should be removed and should not be used for testing. A stock of at least 12 males and 12 females will be needed for routine testing. If the animals will be used on-site, transport them separated by sex in separate or partitioned coolers packed with wet kelp and paper towels. On-site, the sea urchins will be transferred into separate 10-gallon aquarium tanks. Obtaining gametes. Transfer the animals into a shallow bowl filled with enough seawater to just cover their shells. 4 females should yield enough eggs to test 5 test dilutions plus 1 control with 4 replicates. Eggs are obtained from the female sea urchins by electrical stimulation. Touch the shell close to the genital pores with electrodes from a 12-volt transformer for about 30 seconds. The red eggs will pool on the sea urchin's shell above the genital pores. Collect the eggs from the shell using a 10 milliliter disposable syringe with a large gauge needle that has been blunted so that it will rest on the shell. After collection, remove the needle and empty the eggs into conical centrifuge tubes. Repeat this for all 4 females. Keep the eggs at a constant temperature, close to 20 degrees, until use, but not longer than a few hours. All vials, pipettes, and pipette tips used in this test should be soaked in seawater overnight. Obtain sperm from 4 male sea urchins. Like the females, the males are induced to spawn by placing electrodes from a 12-volt transformer against their shells for 30 seconds. The sperm appear white. Collect the concentrated sperm that pools on top of the shell using a 1, 2, or 3 milliliter syringe with a blunted large gauge needle. Keep the sperm sample on ice and record the collection time. Making stock solutions. In preparation for the actual exposure, the sperm and eggs are diluted to constant concentrations with control water. This ensures reproducibility in the test results. In Narragansett, laboratory workers use a standard data sheet for calculating and recording dilutions. Later, during the exposure period, 2,500 sperm will be present for every one egg. After collection, the sperm should be in a volume of about 0.5 to 1 milliliter of control water in the collecting syringe. This is called the sperm stock solution. Perform a 50% serial dilution for counting, keeping all suspensions on ice. To do this, remove the syringe tip and add 400 microliters of the stock to 20 milliliters of control seawater in a vial labeled A. Gently mix the solution by pipetting up and down with a 5 milliliter pipette or by inverting the vial several times. The dilution in this vial is 1 to 50. Take 10 milliliters from vial A and add it to 10 milliliters of seawater in the next vial, labeled B. After mixing solution B, take 10 milliliters out and add it to a third vial, C, containing 10 milliliters of seawater. Vial C now contains a 1 to 200 dilution of the original sperm sample. Repeat the last step once more into a vial labeled D. Discard 10 milliliters from vial D so that all vials contain 10 milliliters of sperm dilution. Next, make a 1 to 2,000 sperm dilution for counting. The sperm must be killed for counting by adding glacial acetic acid. Add 10 milliliters of 10% glacial acetic acid in control seawater to vial C, which contains the 1 to 200 sperm dilution. Cap vial C and mix by inversion. Add 1 milliliter from vial C to a vial labeled E, which contains 4 milliliters of control seawater, and mix thoroughly. Vial E now contains 1/2000th of the number of sperm in the original sperm sample. Now the concentration of the sperm stock can be determined. Place an aliquot of dilution E onto a hemocytometer for counting. Allow the dead sperm to settle for about 15 minutes. Meanwhile, prepare a standard egg dilution of 2,000 eggs per milliliter. Use suction to remove the supernatant seawater from the settled eggs, taking care not to disturb the egg pellet. Add seawater and mix carefully by inversion. Spin the vial on a tabletop centrifuge for the lowest possible setting for 3 minutes to form a lightly packed pellet. Discard the eggs and start again if the wash water appears red. This means that the eggs are lysing and are unsuitable for testing. Repeat this process 2 more times. Transfer the washed eggs to a beaker containing 200 milliliters of control seawater. This is called the egg test stock. Next, make a 1 to 10 dilution of the test stock for counting. Mix the stock solution using gentle aeration until the egg solution is homogeneous. The aeration device used in Narragansett is a 3-pronged diffuser attached to a portable air pump with flexible tubing. Cut the point from a pipette tip to make sure the eggs will not be damaged. Then, transfer 1 milliliter of egg solution to a vial containing 9 milliliters of control water. Mix by inversion. Transfer 1 milliliter of this solution to a Sedgwick rafter counting chamber. Count the number of eggs using a dissecting microscope at 12 times magnification. Ten times the number of eggs in that milliliter equals the number of eggs per milliliter in the egg stock. If the concentration is not between 1,800 and 2,000 eggs per milliliter, adjust the egg concentration to 2,000 eggs per milliliter by adding or taking away some water. Then verify the concentration by counting 1 milliliter of another 1 to 10 dilution of the adjusted stock solution. A test of 5 concentrations and a control with 4 replicates will require at least 25 milliliters of egg stock solution. When the sperm have settled in the hemocytometer, use a compound microscope to count the inner 400 squares of each side of the chamber. At 100 times magnification, the sperm will appear as small specks in the chamber grid. Average the counts from each side of the hemocytometer. This resulting number, when multiplied by 10,000, is the number of sperm per milliliter in vial E. In turn, that number multiplied by 2,000 is the sperm concentration in the original sperm stock. Calculate the sperm concentration in each vial. Then, choose the vial that contains greater than or equal to 5 times 10 to the 7th sperm per milliliter. That vial is diluted to a final concentration of 50 million sperm per milliliter and used as the sperm test stock. Only about 2.5 milliliters of sperm test stock solution is needed for testing 5 test solutions and a control with 4 replicates. Hold the test stock on ice until the test begins, but no longer than 1 hour. Beginning the test. Store the effluent or receiving waters in a 4 degrees Celsius incubator or refrigerator for no longer than 36 hours before first use in NPDES testing and in no case longer than 72 hours. Always maintain the salinity of the test samples at greater than 26 parts per thousand. To do this, effluent samples may need to be adjusted using hypersaline brine. A recipe for hypersaline brine is provided in the supplemental guide. Receiving water samples with greater than 26 parts per thousand salinity may be tested without further salinity adjustment. To test a series of decreasing concentrations of effluent, use a 50% dilution factor. EPA uses disposable glass vials as test chambers. The vials are arranged in the partitioned cardboard box in which they are packaged. Perform a serial dilution from the highest concentration of effluent to the lowest, starting with 10 milliliters of the highest concentration. When the effluent salinity is 0 parts per thousand, the highest effluent concentration possible for testing is 70% effluent. Move 5 milliliters from that vial to the next and so on, finally discarding 5 millimeters from the last vial, which contains 4.4% effluent. There should be 4 replicates of each test concentration and the control solution. This video shows 3 replicates. However, the 2002 revised methods require a minimum of 4 replicates for each concentration and the control. Maintain the samples at 20 degrees Celsius by keeping the vials on the laboratory bench if the room temperature is near 20 degrees Celsius or in an environmental chamber before adding the sperm. One hour after the sperm are collected, add 100 microliters of the well-mixed sperm stock to each test and control vial so that there are about 5 million sperm in each chamber. Cover the chambers, record the time, and leave the chambers at room temperature for 1 hour. Next, mix the egg test stock using gentle aeration and add 1 milliliter of that test stock solution to each exposure vial. When all the vials contain eggs, Lift the storage box and move it in circles to swirl and mix the egg-sperm suspension. Cover the chambers, record the time, and incubate the eggs and sperm at room temperature for 20 minutes. After 20 minutes, end the test and preserve the samples by adding 2 milliliters of 1% formalin in seawater. Again, record the time, then cap the vials. It is best to evaluate the test immediately after the conclusion of the test. The vials may also be saved at room temperature until evaluation, but no longer than 48 hours. Evaluating the test. This test demonstrates the effluent or receiving water's effect on sea urchin fertilization. To evaluate this, exposed and control eggs are examined under a microscope and the number of unfertilized eggs in each test chamber is recorded. Cut the point from a pipette tip. For each replicate, transfer about 80 to 120 microliters of the preserved eggs to a multiple chamber counting slide. If a Sedgwick rafter counting chamber is used, transfer about 1 milliliter to the cell. Using a compound microscope at 100 times magnification, count about 100 eggs. This should be done with adequate ventilation because formalin is harmful when inhaled. For each test chamber, record the total number of eggs counted and the number that were not fertilized. Fertilized eggs are surrounded by a fertilization membrane. Unfertilized eggs lack this membrane. Abnormal eggs are not counted. For the test to be acceptable, the controlled chamber must show 70 to 90% fertilization. The Methods Manual, short-term tests for estimating the chronic toxicity of effluents in receiving waters to marine and estuarine organisms, explains how to analyze and interpret the results of the sea urchin test. The sea urchin sperm cell test is currently used to assess the potential toxic effects of complex chemical mixtures on marine and estuarine organisms. Used in conjunction with chemical-specific methods, this test can provide a comprehensive and effective approach to assessing the impact of complex effluents discharged to the marine and estuarine environments.